Hi everyone, and thank you for joining us today. Welcome to today's Microscopy Masters webinar with Dr. Allaura Cone on the analysis of tetraspanin microdomains on the surface of extracellular vesicles. So before we begin the webinar, we'd like to go over just some quick house rules for anyone who is new to GoToWebinar. So I'm sure you've all noticed on the side, there is a webinar navigation panel on the side of your screen. It contains a few different sections. And if you'd like to more clearly see one specific section, you can simply click on the expand icon, which is located in the top right corner of each of your sections. Today's webinar will last about 45 minutes, leaving approximately 15 minutes for Q&A at the end. You are more than welcome to submit any questions that you have throughout the webinar via the chat section, which is located within your panel. However, Dr. Cone will only answer questions during the Q&A session. And if she's unable to get to your questions, then we will forward her both your question and the email address in which you registered with. And within your navigation panel, you're also going to find a handout section. And here you can find our company brochure. So please feel free to have a look there or on our website located at oni.bio. And please note that today's webinar is being recorded, and within the next few days, you will receive a thank you for attending email with a copy of today's webinar to refer back to. In addition, a video recording of today's webinar will also be stored on the resources section of our website. And now I'd like to introduce you to our presenter for the day, Dr. Allaura Cone. Just a little background on Dr. Cone. Dr. Cone received her PhD in biomedical sciences from Florida State University. She then moved to UNC Chapel Hill, where she currently works as a postdoctoral researcher in Dr. Dirk Dittmer's lab in the Lineberger Comprehensive Cancer Center. And currently, the lab investigates cancers of viral origin, specifically cancers caused by Kaposi's sarcoma-associated herpesvirus. And one goal of the lab is to elucidate the role that EVs play in viral infection, in addition to gaining knowledge on how we can use EVs as a therapeutic. And with that, I will pass the presentation over to you, Dr. Cone, to begin the webinar.
Great. Thank you so much for the introduction, and thank you to the organizers for inviting me here. The cats are probably going to go in and out. They absolutely love it when I work from home. So hopefully nothing bad happens. But I'm excited to be discussing our lab's work on single particle analysis of extracellular vesicles using super resolution imaging. So, of course, first, just a quick disclosure side, which all of these are managed by the University of North Carolina, and most of our funding comes from the NIH. A quick outline of what today's talk is going to be. I'm going to give some background on vesicles in dSTORM and talk about the visualization of extracellular vesicles in both 2 dimensions and 3 dimensions, and talking about tetraspanin microdomains. Additionally, I'm going to talk about using dSTORM for comparative protein analysis. And if we have time, I would also like to talk about this doxosome story that our lab published in 2022. Let me get my laser pointer. Great. So let's get into some background on extracellular vesicles, or EVs, as I will be calling them for the rest of the presentation. EVs are membrane-bound nanoparticles secreted by all cell types and found in all bodily fluids. They're typically distinguished based on their size and method of biogenesis. So microvesicles, as you can see here, also sometimes called ectosomes, are anywhere between 50 and 500 nanometers in size and bud directly from the plasma membrane in a similar fashion to HIV, as shown here. Exosomes are smaller, between 50 and 150 nanometers in size, and are endocytically derived. Invagination events in the lumen of the endosomally derived multivesicular body, or MVB, produce these intraluminal vesicles, or ILVs. When the MVB fuses with the cell surface, these ILVs are then secreted as exosomes, and this process is similar to enveloped hepatitis A virus, or HAV. And as you may have noticed, EVs have significant overlap in both size and biogenesis pathways to viruses. Additionally, EVs and envelope viruses have overlap in morphology. So EVs have a bilayer membrane containing these membrane-associated proteins, which can be similar to the viral glycoproteins. There's encapsulated proteins, which we kind of compare to the tegument here, encapsulated nucleic acids, which are kind of similar to the viral genome. And viral proteins can also modify EVs and package viral proteins. But the main difference between the two nanoparticles are the presence or absence of this capsid. So as described before, EVs package very diverse cargo, which can perform a variety of functions in recipient cells, such as regulation of gene or protein expression, receptor ligand signaling, or metabolic reprogramming. In cancer or virally modified EVs, there can be additional functions in cell survival, migration, immune signaling, etc. One of the defining markers of EVs are these tetraspanin proteins shown here, so CD81, CD9, or CD63. They're involved in the biogenesis of EVs and are often found to be packaged within EVs. Tetraspanins are a group of evolutionarily conserved proteins with 4 membrane domains, a small extracellular loop, and a large extracellular loop. In cell membranes, these tetraspanin proteins were discovered to form detergent-resistant rafts called tetraspanin-enriched microdomains, or TERMs. And these domains not only contain the tetraspanins, which you can see here, but also integrins, MHC proteins, and metalloproteinases. And these terms are important for various cellular functions, including exosome biogenesis and antigen presentation. So before we get into the data, I wanted to quickly explain our purification pipeline for EVs from cultured media. We first obtain the culture media and perform a slow centrifugation just to remove any dead cells. We then use vacuum filtration to remove any debris or large vesicles. And then we use tangential flow filtration to concentrate the EVs using this method here. We then will precipitate the EVs with polyethylene glycol or PEG, and that's done overnight with a rotation. We then will pellet the EVs using another slow centrifugation, resuspend that pellet, and then treat with RNase and DNase just to remove any nucleic acid from the surface. And then finally, we will then run that pellet through a Captocore chromatography system, and that helps remove any protein precipitates from the EVs that might have come down with the PEG treatment. You can see this method published here. But what we really like about this pipeline is that it allows us to generate EVs that have never been subjected to high G forces, such as ultracentrifugation. And this allows the EVs to stay as native as possible, which we can also see here. So we analyze our EVs using cryogenic electron microscopy. And as you can see here, they're nice and round, which has been previously described, and also helps since we don't subject it to those high G forces. We found that our EVs are typically between like 60 and 100 nanometers, which is very much in the exosome range. But what was really interesting with this Cryo-EM data is that we found these areas of high protein density, which you can see here in the orange arrows, along with these smooth lipid dense regions that you can see with these white arrows. So we wanted to look more into this phenomenon, and with that, we employed super resolution microscopy. So just some background, since this is an ONI talk, and I don't know how familiar everyone is. So our version of super resolution microscopy is dSTORM, or direct stochastic reconstruction microscopy. The diffraction limit of light, which is half the wavelength of visible light, is around 200 nanometers. And this means REVs, which again are 60 to 100, are actually below the limit of resolution of conventional light microscopy. So you can't actually determine a single EV from just a group when you're trying to just look under a normal microscope. So what dSTORM does is it uses a high power laser to induce photo switching events of fluorophores, which we describe as blinking events, since they go back and forth between the excited and then the ground state. We then record these blinking events over a period of time in order to reconstruct the image in finer detail, which you can kind of see here. And then here are some more examples of dSTORM. So you can see here with conventional light microscopy, you see some color and really nothing else. However, by measuring these blinking events, you can actually start to see the finer details. So here would be a fluorescence image, and then over time, you measure the blinking events, reconstruct them in order to create this dSTORM image. And then, oh, perfect, it just restarted. So then here's actually a GIF of how it measures and then reconstructs the image over time. So what we first wanted to do is to see what our EVs actually looked like on the Nanoimager using dSTORM. So we label our EVs with a membrane intercalating dye called cell mask green or cell mask deep red, which can be fluoresced by a 488 or 647 laser. So as you can see here, when we image these EVs in 2 dimensions, we have these really nice round EVs. And then when we did size analysis, we found that our EVs were similar in size compared to nanoparticle tracking or NTA, which is kind of the gold standard of size analysis in the extracellular vesicle field. Additionally, we can resolve these EVs in 3 dimensions, which then allows us to start to localize specific regions. So this is kind of an example of the resolution that you can get. So interestingly, when we use the membrane dye, we can actually see that our EVs are hollow, which we call donuts. And this kind of really shows off the power of the super resolution scope. Since EV membranes are really only between 20 and 40 nanometers, and the inner part of the EV, which isn't filled with lipid, shouldn't have dye. And with dSTORM, you can actually see this hollow part, which would be like the proteins or nucleic acids, instead of just the entire thing being filled with lipid. And even though this might not give us quite the limit of resolution compared to Cryo-EM, we can analyze a much higher volume of EVs comparatively. And what's interesting is then you can take that 3D image of the EV and actually start zooming through the Z-plane and making slices through the EV, almost creating like a Z-stack that you typically see in your normal microscope. And we can see here not only the donut hole, but also these patches of higher lipid density. And then here's kind of a video showing what I showed before. So you'll see like this hollow center and then different amounts of lipid or absence of lipid. So this is great. This is all fun. However, there is a barrier. So all lenses, from cameras to microscopes, the highest optical aberration is in the z-axis. So in order to do image analysis in 3 dimensions, we needed a way to reanalyze the data to make it more spherical. So as you can see here, again, when we measure in 2D, so you have your X and Y axis, our EV is nice and round, it looks really pretty. However, when you start to add the Z-axis, you then start to get this elongation. And this is the bell tower at University of North Carolina. So what we can do as a solution is principal component analysis, or PCA, on the EV images. So we establish the origin, and then we'll use PCA to define these directions of variation that you can kind of see here. And it defines the direction of the angle, but doesn't really change the distance ratios. And we use this tool to reorient the data to the axes of the most variation. So this would have a higher variation than this way. And this better understands the underlying structure, and then we can use that to identify clusters within the data set. And then we can cluster those points on the EV surface based on cosine similarity or projecting the data onto a unit sphere, things like that. So here's an example of some of the statistics that our lab did and how the membrane stain can be correlated using cosine similarity. So if the EV was completely random, with the lipid density, you kind of just see small clusters throughout. So you see kind of just random spread of this red and blue. And then in the other way, if it had a polar membrane, we would see similar patterns, kind of similar patterns to the random, but with larger areas of correlation, right? However, we see that these membrane domains within the EV have these really large blue and red squares, which kind of shows, again, another way to see our high versus low density lipid areas, which we also saw by Cryo-EM. So just a bunch of different ways to visualize the same data. But now that we've worked with lipidomains, we also want to work on localizing proteins, specifically tetraspanins, since most EVs contain at least one tetraspanin, sometimes multiple. So this first image is just our two membrane stains kind of overlapped. So CM green and CM red, and you can see in this figure here that there's a lot of overlap. There's some apochromatic error, so there will be a little bit of distance, but for the most part, they're completely overlapped. And then here we have our CM green, so our lipid EV, and then a CD81M cherry protein. And what's really nice about this is you can actually see that the M cherry tag is tightly associated with the lipid. So as you can see here in this model, the fluorescent protein is highly localized on the EV. But then when we use an antibody, which you can see here, so this is a CM red lipid stain, and then an antibody towards CD81, we start to establish some distance, which then can be determined that the CD81 molecule on the EV itself, and then about a 10 nanometer separation just because of the size of the antibody with the fluorescence on the outside. But what I kind of want you to take away from this, and one of the more important findings, is that we always find these tetraspanins on one side of the EV and not really encasing it. Because EVs tetraspanins are known to homo and heterodimerize. So we think that maybe that they're forced together on one side. So then that led us to think about this fluid mosaic model. So what a fluid mosaic model basically describes is that no membrane is ever completely random, and that membrane domains exist. This has previously been shown in cell membranes. As I described before, you have these tetraspanin-rich microdomains existing on the cell surface. But what we want to aim is show that EV membranes too are this fluid mosaic model and not random, and that we can see these tetraspanin microdomains on the surface of an EV, which we want to use a mix of dSTORM and Cryo-EM to prove. So the first method we enlisted is 3-color dSTORM. So again, just going through the colors, we have our cell mask red, so that would just be the EV lipid, and then antibodies towards CD81 or CD9, which are two tetraspanins. And again, as we kind of saw before, the tetraspanins sit on this outside, but they're always next to each other. You don't have, like for this, you don't have CD81 on this side and CD9 on this side. They're kind of always together. So this kind of reproves what we saw in Cryo-EM, that you have this colocalization of tetraspanins or these high protein density areas. And then on the other side would be the smooth area or just lipid dye. And what's extra interesting is that these domains exist independent of purification method or cell source. So in this method, I believe these were U2OS cells, which is an osteosarcoma, going through a normal pipeline of TFF, PEG, and Captocore. And then here we have a breast cancer cell line, MCF7, that was just concentrated using TFF and no additions. And as you can see here, we can see CM red or lipid on one side, and then CDCC3 and CD81 on the other side. And then these EVs were purified from HEK293 cells with differential and ultracentrifugation. So this is including the high G forces that we typically want to avoid. But even with those high G forces, you still see these microdomains existing on the surface. And then we wanted to kind of further look into this, and this is something that we're now working a little more with. So this is kind of early, but we're trying to do 3-color 3D dSTORM, where again in 2D you see the membrane stain on one side and our tetraspanins on the other, but we can also see this in 3D. But you can see here you have some separation because of the antibody, but our tetraspanins are still colocalizing on one side. So just kind of as a result, our lab and others have shown that EVs are enriched in tetraspanins using various methods like flow, nanoparticle tracking, and 2D dSTORM. However, our lab now wants to use the 3D dSTORM to show these specific protein clusters that form these lipid or protein rafts on the EV membrane. So now I kind of want to go to how we use the ONI and dSTORM for other methods of EV analysis. So this paper published by our lab back in June of 2023, we took our normal purification pipeline, but then after the captive core chromatography, which is kind of like a size exclusion method, we then used heparin chromatography in order to separate these purified EVs into two distinct subpopulations, which we call non-heparin binding or heparin binding, which is NHB and HB. So the first thing we used the dSTORM for is size analysis, which I kind of showed previously, where we see a lot of overlap using dSTORM size analysis and nanoparticle tracking, even within the different subgroups. The transmission electron microscopy, the EVs were a lot smaller, but the process of doing TEM also dehydrates them, so there's going to be a little error in trying to compare these. Interestingly, what we found is that the non-heparin binding fraction was enriched in your typical EV markers such as CD81, CD63, which again are tetraspanins, in addition to syntinin or ALEX. And then our heparin binding fraction was enriched in non-canonical EV markers like fibronectin and histone. So we did mass spec, Western blot, but then we kind of wanted to repeat, and it's like, can we also do this comparative protein analysis using dSTORM? So as you can see here with the input, our EVs contain both fibronectin, which is in yellow, and CD81, which is in blue, and then our membrane stain is in magenta here. And then we have our non-heparin binding fraction. We can only really see CD81 positive and not a lot of fibronectin, which you can also see here, 86% of the EVs are CD81 positive, fibronectin negative. And then in the heparin binding, it's the opposite. We now see this fibronectin and an absence of CD81, which you can also see here with 58% CD81 negative. And as you can see with these ends, we can go through a lot of EVs, which creates some really nice statistics. And it just kind of shows the different ways that you can use different methods to validate the same thing. So I don't know how many questions we have about dSTORM. So I don't know if you guys want me to start the story or if you want me to answer any dSTORM questions. But I can stop here if we have a lot.
Let me take a look for you, Allaura.
Yeah.
All right, we do have a few questions. I can read you maybe like 2 or 3 of them, and we'll save the rest for the end.
Yeah, that works perfectly.
All right, someone did ask, how many colors can you use during dSTORM?
Yeah, so technically 4, but the 405 laser itself is kind of difficult. And honestly is usually used more as an enhancer laser for like your 488 rather than using it on its own. So hypothetically, with the right controls and really good fluorophores, you can do 4 color, but we really only find consistency with 3 color.
Okay, and then I'll just take one more that came in. Someone did ask, can you detect viral proteins in the samples?
I didn't show that data, but yes. So we can actually take viral proteins, whether antibody or that are tagged with like GFP, and see them within an EV. However, we've only looked at membrane proteins, so like viral glycoproteins or other membrane proteins. We haven't looked at viral proteins like within the EV, but that's something that we do want to do.
Okay, great. And then I think I'll save the rest of them for for the end. We did just have, maybe we can just take one more if that's okay.
Absolutely, yeah.
Had a question come through. Which buffer do you use for cell mass green red to stimulate the blinking?
Yeah, so we use an oxygen scavenging buffer. So we'd rather use the BQ buffer that ONI sells, or we've also created our own. And that's a mix of glucose oxidase, beta-mercaptoethanol as like a reducer, but just general oxygen scavenging, yeah.
Okay. And then, Allaura, we had 4 or 5 more come through, but I don't want to interrupt the presentation, so I'm just going to let you finish and we'll save them for the end.
Yeah, that's completely and totally fine. This is a lot less dSTORM related, so that's why I wanted to give a chance to answer some dSTORM questions before I start this part of the talk. So back in 2020, our lab published this paper on what we call doxosomes, which are doxorubicin-loaded EVs. Here we go. So kind of to circle back and reorient, that there are basically 3 types of nanoparticles that are all around the same size, around 100 nanometers. So the first are liposomes, which are artificial membrane vesicles that can contain proteins, nucleic acids, or in this case, doxorubicin. Liposome-bound doxorubicin is called Doxil, which is an improved formulation for the treatment of Kaposi sarcoma. Doxorubicin on its own, though, can cause a lot of off-target effects, including heart damage. And then we have the EVs, which we already talked about. They contain nucleic acid, membrane proteins, internal proteins. And then finally, again, you have the viruses, which again contain the capsid that protects the genome. The membrane is usually a lot simpler than an EV because it will be enriched in viral proteins, usually glycoproteins used in uptake. And then a quick introduction to Kaposi sarcoma. So it's caused by Kaposi sarcoma-associated herpes virus, or KSHV, and is a tumor of endothelial cells. It is the most angiogenic cancer in humans, and as I said before, it is treated with Doxil. So as you can see here on the right, the brown parts are the tumor, and then the blue here are the blood vessels. So it's typically thought that what you put into the blood vessels will end up with within the tumor. However, Kaposi sarcoma is also really leaky, so a lot of that can then be kind of like removed from the tumor itself. So our lab has showed that 5 viral microRNAs produced by KSHV will actually account for more than 50% of all microRNAs within a cell. So you can see here just a ton of viral microRNAs, and then this miR-221 and miR-222 are endothelial lineage markers. Additionally, we can show via qPCR the viral microRNAs both in cells and in EVs. So we have our uninfected control here, which don't have the KSHV microRNAs, our infected cell line, and then these were EVs taken from patients. And then we also enriched EVs using CD63, which is a tetraspanin. And you can see here both in our infected cell and in our patient cohort that we're able to detect these viral microRNAs. We also use digital PCR to count individual events. So you can see a lot of viral microRNAs in the cell, of course, but we can also see these microRNAs inside the EV. And we actually found that, and we believe that there's 50 to 100 microRNAs per EV during KSHV infection. So we took these EVs from cells from mock, our uninfected control, our infected EVs, or just infected cells, and we put them, the EVs, onto normal endothelial HUVEC cells, and we found that we could actually, that the EVs alone can induce proliferation, as you can see by this percent of Ki67, at similar rates to the infection itself, which is really interesting. So doxorubicin itself is extremely hydrophobic, so it wants to be incorporated into a membrane. So just incubating doxorubicin with the EV or doxorubicin with a liposome, it will kind of enter in. And conveniently, it is also fluorescent, so you can measure doxorubicin incorporation via fluorescence. So this was actually done via mass spec, though, where we did nanograms of doxorubicin per 1E10 particles. And interestingly, we noticed that the KSHV EVs with doxorubicin actually had more dox present than the liposome formulated doxil. Additionally, we incorporated, we used doxil or the EVs with doxorubicin. And then every day we would run the EVs or doxil through Captacor, which removes any unbound doxorubicin. So when doxorubicin kind of leaves the liposome or the EV, before it can then be reincorporated, we'll run it through Captacor, which kind of removes all of that. And so what we found is that the doxil, clearly the doxorubicin was entering or leaving a lot, because you can see that the retained decreased a lot, even especially after, like, in the 3 to 4 day mark, versus excuse me, the EVs retained a lot of that doxorubicin. We also found that these EVs with doxorubicin actually kill cells by the same mechanism. So we have just regular EVs and DMSO here. So you can see our phospho-gamma H2AX, which is a sign of DNA damage. You see very little of it when treated with normal EVs. And then we have our KSHV EVs with doxorubicin. You then start to see a high amount of this H2AX along with doxil and doxorubicin alone. Sorry, here we go. And then we use an Excelgen system to actually measure how quickly the cells died during treatment. So we have our mock, just EVs with dildi, our EVs with doxorubicin or our doxil, and we found that the EVs with doxorubicin actually killed the cells faster than doxil itself. So in humans, there is an oral form of Kaposi sarcoma that's actually treated by directly injecting doxil into the tumor. So we kind of wanted to see if we could replicate this in mice, where we mixed EVs with Matrigel and implanted it subcutaneously into mice. So when you just have the PVS, mouse cells will kind of migrate into the plug, just because they do. But you don't see a lot of this CD34, which is a marker of angiogenesis. When you add VEGF to the Matrigel, which is vascular endothelial growth factor, you then have a high amount of CD34 positive cells, whether because VEGF is differentiating the cells or recruiting those types of cells to the control or to the gel. And then the viral EVs themselves are also produce a lot of, are very neoangiogenic. But then you can actually reduce the amount of new blood vessels being created by loading these KSHVs with doxorubicin. So what we wanted to do then is determine what actually keeps the doxorubicin in the EVs. So as I previously described, EVs are different from liposomes since they contain microRNAs and proteins. Well, it's really hard to knock out every single protein found in an exosome. That would be a lot of CRISPRs in the cell. So we decided to focus on the microRNAs. So we can get rid of these microRNAs by knocking out dicer, which is essential for the production of microRNAs. So as you can see here, we have our EVs that don't have dicer in the knockout, but are still enriched in our tetraspanin proteins like CD81, CD63, CD9. And then as you can see here, we knocked out the ability for microRNAs to be produced. So as I said before, doxorubicin is fluorescent. So we can actually measure amount of doxorubicin by flow. So we kind of have our negative control here of just DMSO, and then our wild type EVs plus doxorubicin have this nice peak here. But when you knock out the microRNAs by knocking out dicer, we have significantly less doxorubicin incorporation, which we can also see here, where you have a higher mean fluorescence intensity in the wild type EVs compared to the dicer knockout. So this led us to hypothesize that the doxorubicin may be binding to the microRNAs within the EV, as we can see in this conclusion here. So doxorubicin, like I said, will enter the liposome, but can also leave the liposome kind of just as easily. However, in an EV, it will go into the EV, but when it binds up the microRNAs, it can no longer then leave the EV. So we think that the presence of these microRNAs is really helping with the phenotypes that we previously saw. Now, we didn't completely ablate the ability of doxorubicin to not leave with just knocking out the microRNAs. So there's probably some other things present, like proteins or other nucleic acids, like tRNAs within the EV that the doxorubicin might be binding to, and that's something we want to look into in the future. So with that, I would like to do my acknowledgements. Huge thank you to the Cryo-EM Core and Dr. Griffith with helping us with transmission electron microscopy, our funding, our bioinformaticians, Justin and Alice, who are so patient when it comes to me asking them 1,000 math questions, in addition to everyone else who is part of Team Exosome. And with that, I can take more questions.
All right, wonderful. Let me just share my camera again so I can come back on with you. All right, great. So thank you so much for the presentation, Dr. Cone. We're now going to open the floor to questions, which Dr. Cone will be answering live. And once again, if you do have any questions, please just feel free to drop them into the chat box. It looks like we do have some questions submitted already, so let's go right into the first one, if that's okay.
Sounds
so
great.
We did have someone ask before, what do you need to do to compare samples from different sources?
Yeah, so I'm assuming they're talking about the dSTORM EVs. But we have these Abiti 8-well glass chambers. So it's a 1.5 H cover slip, which is the like glass you need for the diffraction. And we can actually do like of course, your antibody only controls and your negative controls, but directly compare the different EVs taken from different cell types. And then from there, when we collect the data, we'll actually upload it into Cody, which is the cloud-based image analysis software that ONI uses. And then from there, we can do different types of analysis based on like percent positivity, radius, stuff like that.
Wonderful. All right. We had someone else ask for Dr. Cone, can you track EVs as they are secreted or received by cells?
No, that's a really good question. Our lab personally has not tried that, but the answer is yes. So there's this nice fluorescent molecule, I think from Alyssa Weaver's lab over at Vanderbilt. It's called CD63 phalloidin. So when it's inside the cell, inside the MVB, the pH is a lot lower. And then when it gets secreted as an exosome, it will then fluoresce because the pH difference. So it's phalloidin with a pH. And from there, you can then track exosome secretion since you know it's being developed from the multivesicular body endocytically. And then for uptake, our lab has done those experiments where we will stain EVs with CM red, CM green, depending on what other markers you want. And then you can incubate those EVs onto cells and then measure just general fluorescence of your membrane stain via flow or IF or dSTORM.
All right, wonderful. We had another question. How can you tell which cells have which tetraspanin?
Yeah, so tetraspanins themselves are found on most cells, though at different ratios, right? They're involved in a lot of cellular processes, depending on what tetraspanin it is, but most cells have the three main tetraspanins that I at least study, which is CD81, CD9, CD63. You can do like Western blot or IF to determine like different ratios of what, like the presence of those tetraspanins within the cell, and then kind of correlate that to your EV data too. But hopefully that answered that question.
All right, great. We did have someone ask, and then maybe Shaked, you can write this out for them. If you can just comment again, Dr. Cone, on the buffers that you used.
Yeah, so the main buffer that we use for EVs is rather the BQ buffer that we get from ONI, or a general oxygen scavenging buffer. I don't remember what paper we got it from, but I'm more than happy to email. I have my email listed here. But it's glucose oxidase, catalase, beta-mercaptoethanol, tris. And I think that's it. And then we also, we've done some cell dSTORM, and we'll actually use a glycerol-based enzyme-free buffer, just because the enzymes will degrade over time, and then so your imaging can only last so long. And we've tried things like Prolong Gold, but we're still kind of testing that, so I don't have a great answer for that quite yet.
Okay. Right, we had another question come through. They said, have you tried this method to study cargo proteins in EVs? And I think they're referring to dSTORM.
Yeah, we've started. So the non-membrane bound EV proteins within an EV are a bit harder to study. So because you can't just put an antibody on the outside, and then your Alexa fluors are really great photo blinkers, and you can get really nice signal that way. When you're looking at internal cargo of an exosome, you have to use like a GFP or an M-Cherry, which are a lot worse photo blinkers. So we're still kind of doing that data, and that's one of the reasons finding those donuts within the 3D imaging is so critical. Is then you can see, you know, your membrane on the outside, and then hopefully like a GFP signal on the inside of like the hollow EV, right? But that's still very early, and we're hoping that we can get better resolution soon.
All right, wonderful. We had another question come through. Someone said, how well corroborated is that EVs have a toroid form? Could some sort of artifactual photon lensing effect be occurring to drive detected photons to the periphery of the EV in the XY plane?
Yeah, okay. That's a really great question. So typically in order to see the toroid donut, the EVs typically have to be a little bigger. So if you have like a 50, 40 nanometer EV, the whole thing's just lipid, right? It's when you're starting to get a little bigger, that the membranes are physically farther apart, that then you can get that hollow center. It is a good point. There is some issues when it comes to any artifacts that you can find just doing microscopy. But that's one of the reasons we try and take it out of the Cody system and start using like Mathematica and R in order to kind of alter how we physically analyze that data. And I think a lot of those equations were published in, I believe, the McNamara JEV 2022 paper, and also all equations are made public on GitHub, I believe. Yeah, if you wanted to look more into that. I would also be happy to talk more about that when I have a math person present.
Okay, wonderful. Thanks so much. We have a few more questions coming through. So next one is, all right, so someone asked, I have M neon green tagged cargo proteins within a particular subset of EVs. I'd love to do dSTORM. Can M neon green photo blink? You just mentioned GFP as a worse photo blinker.
I don't specifically know about neon green. Personally, I've never worked with that. GFP does blink. It just doesn't blink as well as like your Alexa Fluor antibodies. So I'm not sure. Usually you can Google the fluorescent molecule with photo blink, and it usually tells you. But I personally have never worked with the neon, so I'm not sure.
Yeah, no problem. They also commented, some sources say yes, some sources say no, so they were just curious what you thought of it. Okay, so someone said, can you comment on differences in throughput and sample preparation between SuperRes and Cryo-EM?
Yes. So we have a really great seeding and staining protocol, which was published in that 2023 JEV paper for heparin, the newest protocol, that we can do really high throughput. Like in a day, I can get like 5,000 to 6,000 EVs per like 4 different samples, and then do statistics, right? Versus Cryo-EM, the actual procedure to incubate the EVs onto the grid and then doing the flash freezing and liquid ethane, I believe. Sometimes we get really great grids where the EVs are within where you can actually see them and they're not too icy. But then we have other grids that just the EVs get destroyed, or they're not necessarily where there should be. They're in like the very carbon dense areas, so you can't really get good resolution. So that's something we're working on for cryo. But I think if you have a good seeding protocol, especially for dSTORM, you can do a lot more EVs a lot faster than you can typically with Cryo-EM.
All right, wonderful. We had someone ask, how common are non-Tetraspanin markers in EVs? And have you ever tried STORM with any other surface markers?
Yeah, so we definitely do see a percent of EVs that have no tetraspanins, period. So there are the surface markers. There are EVs that don't have any tetraspanins. That just is well described in the field as kind of being an enrichment marker, which is why we focus on those. Being a virology lab, the other membrane proteins that we've looked at usually are viral proteins. So glycoproteins associated with KSHV or membrane-bound proteins of different viruses. We've done a little work with like coronavirus spike protein and looking at that on the surface of EVs, which hopefully that paper is also going out soon. But yeah, you can, I mean, I think most, as long as there's a good antibody for it, you can usually look at it.
Great. Right on to the next question. Someone asked, what is the minimum number of localization you have set in your analysis for an EV to be considered positive for that specific stain?
Yeah, that's a great question. We actually have a table also in that JEV heparin paper with all of our parameters we set in Cody. So please feel free to look at that. I believe it's 30 localizations though, for it to be considered real.
Wonderful. Next question, someone said, hi, thanks so much for the great talk. Is there any particular reason why you use TFF PEG and CaptoCore 700 isolation sequence? And could you please comment on the utility of each?
Yeah, that's a great question. So our lab in particular is interested in using EVs as therapeutics. So without having to use ultracentrifugation, excuse me, and things like gradient centrifugation, in order to get purified EVs, you can scale up a lot easier. So we actually worked a little bit with a bioreactor. I think I actually, yeah, we actually worked a little bit with the bioreactor where we have this huge amount of media that we then can run through TFF and concentrate it down to like 50 or 100 mils. And we can produce 10 to the 13 EVs per week at 10 to the 11th per mil. So let me find my cursor again. Here we go. So that's kind of one of the reasons we did it, was mostly because of scalability, which you cannot get when you're relying so much on centrifugation. And a lot of the PEG and CaptoCore actually were originally used in purification of viruses. And since EVs and viruses have a lot of overlap and we're a virus lab, we kind of stole from the virology field, stand on the shoulders of giants, and just kind of repurpose that for our uses.
Great. And then to follow on to that, someone said, in lieu of your answer, have you ever tested EV uptake contrasting different isolation methods?
That's a great question. Yes. I don't remember if that data is published or not. And I also sometimes I don't remember what I did in my PhD versus my postdoc. But yeah, we have done some uptake experiments with different methods of purification. We personally, I don't remember finding any significant differences between them. The main difference is just that the TFF method is just much more scalable, so you can get a higher ratio of EVs in a week compared to trying to do it by ultracentrifugation.
All right, that might be all the answers for you. So I think that's all the questions we are going to take for Dr. Cone for the time, and it seems that we're very close to time. So thank you again for the presentation, Dr. Cone. And if anyone has any questions that they still want to ask, you can still ask it here or in the survey afterwards, and we'll forward them over to Dr. Cone to answer. And then once again, this webinar was recorded. Therefore, if you need to refer back to it, you absolutely can. And all these recordings can be found within the resources section of ONI's website. So make sure to check that out. And a big thank you to all the attendees as well for joining us today. Upon exiting the webinar, you will receive a survey. Please fill out the survey to provide us feedback on your experience today, or to tell us if you're in need of being contacted directly by ONI, or if you have a question for Dr. Cone. And thank you again, and we hope that everyone has a wonderful day.
Thank you.